Hello, my name is Ben van den Wouwer and I'm a PhD student at Research Unit VEG-i-TEC of Ghent University and the Laboratory of Food Chemistry and Biochemistry of KU Leuven. Did you know that 1/3 of the food produced for human consumption is wasted along the food supply chain? This is a pity because we can still recover proteins from these food byproducts and reintroduce them in the food production chain. And this is exactly what I focused on in my research. More specifically, I studied the recovery of proteins from potato trimmings, which is a byproduct of potato fries production. The end goal is to use these proteins as a replacement for animal proteins in foods. In other words, how we can go from a low-value byproduct to functional ingredients that can be used in higher-value applications. Typically, during a protein extraction process, the proteins are extracted by mixing the potatoes with water. After this, they are isolated again from this water. But this extraction process has some challenges. A first challenge is that plant proteins are located inside of plant cells, making it difficult to extract them. This often results in low protein recoveries. Secondly, the extraction process we use has to destroy these cellular structures, but it cannot destroy the proteins because they have to remain functional so that we can still use them. And thirdly, all of this has to be done in a cost-effective way. Luckily, we discovered that by using a mild extraction process, we can isolate potato proteins that have a similar or even better foaming properties compared to commercial potato proteins on the market today. Additionally, we found that novel technologies like ultrasonication, enzyme treatments, or bead milling could efficiently break down the potato cells to increase the extractability of the proteins. Moreover, if we combined ultrasound treatments with enzymes or bead milling, we found that the protein recovery could be additionally increased. Another focus of my research was the translation of processes that work for potato trimmings to other byproducts. And we found that while some processes were translatable to other matrices, this was not so easy for others. And therefore, it seems that one technique does not fit all. And this is because each plant matrix has a unique composition and structure. But this also opens new possibilities, because each byproduct also contains a unique set of proteins, and these proteins can all show different functionalities. So while their extraction might be complex, the diversity of the proteins in these byproducts will be of great use, giving the wide range of applications for plant-based proteins. So what my research shows is that by making clever choices while setting up extraction processes, we can recover functional proteins and increase the value of those food byproducts. And this all contributes to a more circular economy and hopefully less food waste in the future.